Hi, and welcome to another episode of Tom Kennedy's Science. And I'm your host, Dr. Tom Kennedy. Now, this lecture is part 5 of my series on the cell. Now, part 4, just before this, was on the parts of the endomembrane system. We went through the laundry list of all the different components and organelles that form the endomembrane system. Now, today, what I want to talk about is how the endomembrane system works as a whole. And we're going to focus on two things. We're going to talk about how proteins are exported outside the cell, and then we're going to talk about how the cell recycles materials. But let's start with the first one. If you're like me, you like pizza. I mean, I like pizza a lot. I know I talk about food all the time. You're going to get some pizza in because you're hungry, with like some green chili, some jalapenos, and some bacon. I live in New Mexico, so I like my peppers. And of course, you know by now that green chili is seriously the bacon of the vegetable world because, green chili goes well with everything, right? But I digress. You get that pizza, you smell it, and you're hungry, right? So you almost have this Pavlovian response to start to salivate. I think I'm salivating now. Yes. Well, your saliva is an interesting thing, and it has these protein enzymes in there, and one of them is called amylase. And amylase starts to break down starch. So as you're chewing your pizza, not only are you breaking it down mechanically, you're breaking it down chemically as well. But you should ask this question. We know that proteins are made by ribosomes inside the cell. So how do you make a protein inside the cell and then export it outside the cell so it's in your saliva? Well, if your guess in the endomembrane system, you're correct. Let's take a closer look here. And what I want to start with, of course, is the big picture. All right, there's a few steps here. This is it in a nutshell. You smell. There's little pieces of pizza floating around in the air. They bind to your olfactory bulbs, and your brain interprets that and goes, yum, pizza. Your stomach communicates with your brain. You're hungry, man. So, the brain then sends a signal to your salivary glands, so you begin to salivate. And in doing so, you start making the protein amylase. And it works like this. The signal gets to your salivary glands in your mouth, and then it receives that signal. And then the signal gets, transmitted inside the cell where it gets into the cell nucleus. And it begins a process called transcription. Now, transcription is where you copy the information off your DNA onto a messenger RNA. Because you know that the DNA is what holds the information to make amylase. But the DNA doesn't make the amylase, right? So you transcribe the information. It's like copying your notes, right? Into a messenger RNA. Then the messenger RNA exits the cell nucleus through the nuclear pores. And it reaches the cytoplasm. And then once it reaches the cytoplasm, it binds to a ribosome. And then the ribosome will make its way with the messenger RNA down to the rough ER, where it will then translate the protein inside the rough ER. Now, translation is where I take the information in my sequence of nucleotides and translate that into a sequence of amino acids. Translation, like translating Spanish to English. You get the idea, right? And then what happens is you make a protein inside the rough ER. It gets into a transport vesicle, moves to the Golgi body where it's processed further, and then it leaves the Golgi body and then is exported to the outside of the cell. That's it in a nutshell. That's big picture. But if you're like me, there's more to this story. There's always more to this story. So let's take a closer look, because I know I have a question like, well, once your messenger RNA binds to your ribosomes, how does it make its way to the rough ER? Well, let's talk about this. So ribosomes, these are large molecular complexes that are the site of translation. And translation is also known as protein synthesis. I'm making proteins. So your ribosomes are floating around in your cytoplasm. Actually, they're in the large and small subunits. But once it binds to the messenger RNA, it immediately comes together and translation begins. I'm starting to form a polypeptide. Many peptide bonds, proteins, right? So all proteins are polypeptides. Polypeptide, of course, is just a sequence of amino acids, but a protein, well, that's an actual folded polypeptide that's biological function. So we start translation. Now the issue here is translation stops, at least temporarily. Because what happens is the first few amino acids that are translated, well, they form what is called a signal sequence. And that binds to a protein called the SRP for signal recognition particle. There's a protein that binds to it. And when it binds to it, it stops translation, at least temporarily. And then the ribosome is bouncing around the cytosol, and eventually the ribosome will make its way to the rough ER. And on the rough ER, there are signal recognition particle receptors, SRP receptors, right? And that allows the ribosome to dock to the rough ER. And once this happens, it docks, the SRP, the protein that stopped translation, it pops off. You pop that off, guess what? Translation begins again. And what happens is, as you start to translate your polypeptide, it gets pushed into the rough ER through the little pore opening called a translocon. And then as you have your polypeptide is being formed inside there, it gets folded into a protein. And something like amylase it gets folded into its tertiary structure. And inside of the rough ER, it's kind of cool, there are these other proteins called chaperonins that help fold your polypeptide into a functional protein. Now also what happens is that signal sequence, that's not actually part of the protein. It gets cleaved and then is removed. Now, while the protein is being made inside the rough ER, it gets tagged with these oligosaccharides. Another big word again, right? Oligo just means small, saccharide means sugar. So you put these short chain sugars onto your protein and you form what is called a glycoprotein. And those have the purpose of telling the cell where to send that protein. So in this case, they're going to go to the ends of the rough ER and they're going to form a transport vesicle and they're going to move to the Golgi apparatus. Now, in my final series of this cell, you'll find out that these transport vesicles, they don't randomly float between the rough ER and the Golgi. They actually walk down the microtubule tracks. It's kind of cool. So at any rate, they get to the Golgi apparatus. Now, the Golgi apparatus is like a series of flattened sacs, like pancakes. I know, another food reference, but that's okay. And each of these sacs is called a cisternae. And they have a cis side and a trans side. So what happens is the vesicle will merge with the cis side of the first cisternae and release a protein in there. And what the Golgi body is going to do, Golgi body, Golgi apparatus, you can call them either one, is going to modify that protein. It might activate it, it might finish adding some foldings to it, it might add other sugars to it as well as additional tags. And what will happen is that the protein will move through each one of these cisternae and have an individual modification done. So it's like a series of steps of modifications to this protein. And then eventually, when it gets near the end of the Golgi body, the protein will then form another transport vesicle, and because it's tagged, it will go to the cell membrane, where then it will merge, the vesicle will merge with the cell membrane, and then it will release it outside of the cell, and that's called exocytosis, and it means out of the cell. Now, the Golgi body doesn't just process amylase, it processes hundreds of different types of proteins and will form these transport vesicles that are destined for many different places within the cell. So, guess what? There are small receptor proteins on there that guide these vesicles as to where to go. So, for example, they could also go to a lysosome, because a lysosome is involved in cellular digestion, right? You got all these digestive enzymes, and you definitely don't want them floating around the cytoplasm because they break down all the molecules in your cytoplasm. It's kind of like your stomach, right? You don't want your stomach releasing all of your digestive enzymes into your body cavity. That would be really bad. It would actually kill you. So now let's talk about the lysosomes and recycling inside the cell. Now, they have 50, 60 different types of enzymes, like nucleases and proteases and lipases. And there's different ways they do this. They can do an endocytosis, which is usually small things, phagocytosis, which is a type of endocytosis, but for larger stuff, and then autophagy, which means self-eating. So let's take a closer look here, starting with endocytosis. One particular type of endocytosis is called receptor-mediated endocytosis. Okay, if you remember, cell membranes are like 50% by weight proteins. And some of these proteins are receptor molecules. And if they bind to another type of macromolecule, it will induce the cell to do endocytosis, where it'll take the cell membrane and literally wrap around the molecules and form what's called an endocytic vesicle. And then that vesicle will actually start pumping protons in there, right? It lowers the pH to help start with digestion. And then another vesicle coming from the Golgi will then merge with that. You know, these vesicles are like soap bubbles, right? They easily merge together, releasing all these enzymes in there and probably activating them, and it will break down those molecules. That's how you form a lysosome. Now, phagocytosis, like I said, that's another form of endocytosis, but the difference here is this is for larger materials like parts of a cell that may be like a bleb from an apoptotic cell. Cells can kill themselves and they form blebs in the process, which will be taken up by our immune system. They could be bacteria. It could be even cancer cells. So phagocytosis is defined as taking in much larger objects. And the cell, once again, will take its cell membrane, wrap around the object, and by doing so, it will form what is called a phagosome for phagocytosis, and then the some means body. And then a lysosome will go up and merge with it and release all of those digestive enzymes like lipases, which break down lipids, proteases, which break down proteins, and nucleases that break down nucleic acids. And the last one is autophagy. Now, auto means self, phagy means feeding. Cells actually turn over their organelles. And if you remember from an earlier lecture, mitochondria only last about 10 days. So over time they age, they become damaged, they become less efficient. So our lysosomes, they will go and merge with these mitochondria and digest them and turn them over. Now, this is really important for our health. It turns out that as we age, we run out of energy. I mean, we see this, or we don't run out, but we see that we have less energy. Look at a little kid versus the grandparents, right? The kids are all running around in circles. They're quite kinetic. And the grandparents are sitting in the chair watching the kids run around. Or you've got a puppy and an old dog, right? The old dog is sleeping. The puppy is just running around generating entropy. Sorry, I couldn't help myself. Little entropy generators, right? They're agents of chaos. If you've been around little puppies, they're just so full of energy. But I digress, like why do younger animals have more energy than an older animal? Okay, there's lots of reasons, but one of them has to do with autophagy. You see, as we age, the rate of autophagy slows down in our cells. That means we accumulate older, more damaged, less efficient mitochondria. You're producing less ATP. If you're producing less ATP, you have less energy. So recently, scientists learned how to reset the rates of autophagy to a more youthful state. They're kind of resetting the clock on these cells. Now, there's lots of reasons why cells age. This is one of them. This is one part of the puzzle. So imagine if we could reset the clock on autophagy as we age. We could maintain higher levels of energy later in life. So we're getting more exercise, we're having less muscle wasting, less cardiovascular disease. We live a longer, more full life. So that's exciting to me. All right, well, this has been another episode of Tom Kennedy's Science, and I hope you enjoyed it. Now stay tuned for my last episode on the cytoskeleton.